See, for the lysis of the cell, it is very important to break down the cell membrane. And uh, here, a buffer is used. I will tell you, we are using two things, tris and EDTA. EDTA binds with the divalent cations such as calcium and magnesium. Thus, actually, this calcium and magnesium are required for stabilizing the membrane. When calcium and magnesium will not be present, the stability of the membrane will be lost, and finally, the membrane will be destabilized, and it will be easier to break down such a destabilized membrane. Okay, similarly, tris is the main buffering component. A buffer is needed. Why? Because a buffer is actually maintaining the pH of the buffer, maintaining the pH of the whole experimental soup. We'll call it an experimental soup because it is having all the contents of the cell. So the pH can be altered. pH must be altering with the action of the enzymes, and like the same way we are using buffers in the fermenter in class 11th, I told you, yeah, right? Similarly, here also a buffer has to be used because continuously some kind of metabolic activities, some kind of reaction is going on inside the reaction mixture. So to maintain the pH, buffer is very much important. So for now, just putting everything together, lysis of the cell will require EDTA, and EDTA is very much necessary for breaking down or destabilizing the membrane. And these tris will be acting as a buffer, to maintain the pH. pH will not be regulated. If the pH will change, then the cell may denature, and the DNA inside it may also denature. We don't want the DNA which we need to extract to be denatured. We want it in a particular, in a preserved manner, in a very good condition. The DNA of insert should be in a very good condition, and we thus want to prevent it from any kind of disintegration or any kind of problem happening to the DNA. So that is why we are using tris and EDTA. Next step will be stabilizing the DNA. Once the DNA has been exposed, see, when we are using ADTA for a longer duration of time, it will break down the cell membrane as well as the nuclear membrane, and the DNA will be exposed. So once the cells are broken apart, their DNA and contents are exposed. And additionally, we are adding here RNase enzyme. See, in stabilization, once the cells are broken apart, their DNA and contents spill into the buffer. Additionally, what we have added? We have added RNase, which destroys the RNA because we do not require RNA. What we require is the DNA. Second is proteases. We are adding proteases to destroy or digest all the proteins which are present. That is also of no use for us because what we are interested into are the DNA. Other thing which we will add is the SDS, sodium dodecyl sulfate. It is a very important component because it solubilizes the membrane fragments. The membrane has been already broken down by the EDTA, but it needs to be digested. So that solubilization of the membrane fragments are done by SDS. They will break down the fragments into smaller fragments so that if these membranes are not attached to any of the important components which we need, should not be attached to the DNA in any case, okay, and should not interrupt the reaction process further. Our experiment should not be damaged by these fragments, so we really need to remove them. So then we are using SDS. Then taken together, this soup of the cellular content and fragmented RNA and proteins can have a big impact on the pH of the solution. Of course, when what we have done, we have added too many enzymes, proteases also, RNAs also, and then we have added sodium dodecyl sulfate also. So these together, all together, will change the pH of the solution. And once the pH of the solution is changed, this may affect the property of the DNA because DNA is a pH sensitive component, and it is very much important to maintain the pH of the solution. Otherwise, the DNA will be degraded. So what we will add? We have already added tris. Again, some more amount of tris can be added. I'll give you that in a other sequential order. Clear? So for stabilizing, what we are doing, we are removing the proteins, we are removing the RNAs, we are adding sodium dodecyl sulfate for the removal of the membrane fragments, plus we are adding the tris to keep the pH normal, to maintain the pH normal. Next step is DNA precipitation. What we have done till now, we have lysed the cell, we have stabilized the DNA. Once the DNA is stable, we need to precipitate it. This is the final stage of DNA extraction, and to extract it from a solution, DNA is made insoluble by adding ethanol or isopropanol. DNA should be insoluble. It should not solubilized in the SDS or TRACE or EDTA. It should not become affected by any of these components. So what we are doing, we are adding chilled ethanol here. When it is done, the DNA becomes obvious in the solution as white thread. They are revealed. They become visible in the solution. This experiment, once the school reopen, we'll be doing this experiment